So last up, we have Mihaela van der Schaar, who's a professor at Cambridge University, and she will tell us about how AI is contributing to the next step of drug development.
Thank you very much. Thank you very much for the kind introduction and for the opportunity to speak. We have heard about some fantastic ways in which machine learning can enable us to design new drugs. But as Gisbert was saying, are these drugs indeed going to be useful for a particular patient given their unique characteristics? Today, I'm going to talk about how machine learning can enable us to learn from observational data which drug is good for which type of patient, given their characteristics. I'm going to, in view of time, just very briefly introduce some key ideas, challenges, and solutions. So the problem is easy. It's easy to pose, but not easy to solve. We would like ideally to identify what treatment or intervention is best for a particular individual that suffers from a particular disease. For instance, we have here a patient that was just diagnosed with breast cancer. And the question may be, should we give after surgery to this particular patient chemotherapy or not? And if we do, what type of dosage and what type of compounds to use? At this stage, you may wonder whether randomized controlled trials, which are the golden standard, could be a solution to this problem. But as we know, treatment effects are often heterogeneous, and they vary across the different patients. And in fact, randomized controlled trials are not able to solve this particular problem because they are small sample size. They are not representative in terms of the patients that they enrolled. Many of these patients may not be elderly or may not have the comorbidities like the representative patients do. Also, some medication may not work over time, and one may need to change to other types of treatments over time. And they do not capture the complexity of care. And in some cases, they are even not possible to run due to ethical issues. So an alternative is to learn from observational data, which comes in forms of electronic health records, clinical registries, and other such sources of data. They are large sample size, and they do contain representative patients, and they do capture many aspects of care. And if we would be able to do that, they will enable us to do inexpensive and fast understanding of which treatments would be best for a particular patient, given unique characteristics and unique healthcare situation they are going to be treated in. But of course, doing that is tricky because this requires estimating individualized rather than average treatment effects from observational bias data. And this is tricky because both of missing counterfactuals as well as confounding. So the type of problem we would like to solve is to learn from observational data that contains patient features, potentially patient features over time, treatment assignment, which is biased because it is based on actual clinical practice, as well as outcomes of patients over time. And in this case, of course, this is not a randomized controlled trial. The clinicians are not closing their eyes when assigning these treatments. So we need to learn in the presence of such biases. And we do not have counterfactuals. We do not know what would have happened if these patients would have been treated with another drug or would not have been treated at all. What would have happened if they were treated with another dose or at a later moment in time? And this is a very complicated machine learning problem. In fact, much more complicated than any translation problem or than playing games of Go. It's a very complicated causal effect inference problem. And again, we do not have labels. We only have labels for what we have done and what we have observed. and not for the many other counterfactuals. So one can pose that as a causal inference problem, where we are learning from observational data using machine learning. Machine learning can help in this situation in the following way. Due to their flexibility, we are able to model in a very flexible way with a variety of architectures the treatment assignment variable W. Also, we are able to deal with selection bias by crafting different types of losses to train various architectures. There has been tremendous progress in the last 6 years on this problem of learning individualized treatment effects from observational data. Early progress was around 2016 from MIT, and then you see a variety of works, and our own lab has done a significant amount of work to solve problems in binary treatment effects, multiple treatments, continuous treatments such as dosage, treatment effects over time, and many other settings. If you want to learn briefly about this topic, we recommend a tutorial which we have written for a clinical pharmacology and therapeutics journal recently. Also, we have a very diverse set of resources for anyone who is interested to learn about these cutting-edge technologies. Please take a look. Today, I'm going to just very briefly show to you one particular example which we developed with clinicians at the University of Amsterdam Medical Center, as well as in Cambridge, where the focus was to combine ideas from pharmacology with ideas from machine learning. And what we want to show to you here is that machine learning, in joining forces with biology and pharmacology, can be significantly empowered. The problem we wanted to address was the use of dexamethasone for COVID-19 patients in the ICU. And this is a very complicated problem because it requires managing the level of immune response, which is challenging because the right level of immune response varies across the patients. Because the immune response is not directly observable in clinical settings, clinicians often resort to unspecified inflammatory markers such as the C-reactive protein, or for short CRP. The PKPD model of dexamethasone is actually very well understood by the pharmacologists. And better markers of immune response, such as cytokine type 1 interferons, can be measured, but they can be only measured in the laboratory setting. And they are included in the pharmacological model. However, they are not easily measured in the clinical setting, and therefore they are not available to the clinicians. So the question was, how can we use these powerful pharmacological models together with machine learning to empower clinicians? So we created a model, a machine learning model that enables us to bridge the gap between the research lab, the pharmacological lab, and the clinic. Pharmacological models describe the dynamics of these carefully medically chosen variables in the form of an ordinal differential equation. However, as I mentioned, these models describe a limited collection of expert variables, which I'm going to call physiological variables, which are not observed in clinical environments. And these pharmacological models are typically developed and calibrated in the laboratory, and they are not really based on expert variables which we can directly measure in the clinic. On the other hand, in clinical environments, the physiological variables are frequently not observed. Instead, we observe clinical biomarkers and endpoints, and they are important for clinical decision-making, but they do not take these PKPD models, these valuable PKPD models, due to their high complexity and the unobservability of many of these parameters. So these two worlds of pharmacology and of clinical decision-making are often not interacting. Using machine learning, we are able to create this bridge between the research lab and the clinic. And we are doing that using a machine learning model that we have built that we call a latent hybridization model. And this embeds a given pharmacological model into a larger latent variable model, a system of neural ODEs. I'm showing here an example of one particular pharmacological model, but the reality is that you can use many others. And also, while I'm showing here an example from pharmacology, you can also embed other settings, for instance, from physiology, from biology, or epidemiology. So you can take an expert model that you have developed in the lab and embed it in a similar way in a machine learning model. Note that in this latent hybridization model, we are able now for the first time to use observational clinical data to learn both the evolution of the unobservable latent variables and the relationship between the measurements, the clinical measurements, and all the latent variables. And these latent variables include both expert variables from the pharmacological model, as well as other latent variables about the state of the patient, such as, for instance, their comorbidities or other physiological measurements. Note that the practical extensions of this latent hybridization model could include static covariates, thereby enabling personalization. And also we can model informatively sampled data, which is very important in clinical settings because we do not have observations at all times. Finally, in the current work, we also assume that the pharmacological model may be incorrect in several ways. The obvious way in which this functional form of the pharmacological model may be wrong is that it's misspecified. For instance, it uses a linear model, while in effect, we may have a nonlinear model. And many existing techniques can address such misspecification, and they are integrated into this latent hybridization model. We are learning using a very elegant Bayesian variational inference model, the parameters of this latent hybridization model. Then we have used this extensively together with clinicians in the University of Amsterdam hospital. And we have used a model to infer the expert variables for the immune response to viral infection, as well as dexamethasone concentration, information that was not possible before, and then show to the clinicians how the pharmacological model actually results in the biomarkers that we expect to see in this particular patient, thereby enabling them to make better decisions. In addition, I want to show something else that is interesting. We know that machine learning models are usually not very smart. And if they have only limited data to learn from, which is often the case in medicine, unlike in the previous two examples where we have the advantage of many, many examples, in the clinical settings, we may have very few patients only from which we can learn from. In COVID, in the ICU, initially, we had only very few patients. And machine learning models alone, for instance, very powerful machine learning models developed, for instance, for natural language processing or many other settings, were incapable to learn effectively when we had 100, 200, or even 1,000 patients. However, these models, which are learning not only on the basis of the available data, but also the knowledge of the pharmacologists, were able to learn much more effectively. What you see here is the precision accuracy associated with a variety of predictions, and the lower the number, the better. And what you can see is that this latent hybridization model, which combines the already available knowledge from pharmacology with the machine learning model information, this hybridization leads to significant performance improvements than conventional machine learning models alone or expert models alone. Finally, I'm presenting here just very, very little of what can be done using this type of models. If you are interested to learn more about machine learning and what it can do in healthcare, We have an engagement session that we call Inspiration Exchange, where we are trying to join forces with machine learners interested in healthcare from around the globe. And we have a next session on September 19 at 4 p.m. UK. So if you want to hear more and delve further into this type of methods, please join us. Thank you very much.